Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the Deep Dive, where we slice through complex scientific research to deliver the essential knowledge you need.
Today, we are exploring something really fundamental.
It really is. It's this surprising idea that our ability to learn from rewards, the absolute core of motivation, isn't constant. It's not stable.
It actually fluctuates. It moves in a rhythm governed by our own biology.
And if you're someone who likes to get up to speed on these big topics fast, this is for you. Our mission today is to dive into a really elegant study from Nature Neuroscience.
And it's all about the molecular link between the hormone 17-beta-estradiol, which is the main, most potent form of estrogen, and the brain's primary reward signal, dopamine.
It really is a beautiful piece of detective work.
I think so. And the core finding, well, it's pretty compelling. Estrogen doesn't just, nudge behavior a little bit. It seems to naturally amplify the reward signals inside the brain's motivational center.
Which means what exactly? Better learning.
Exactly. It leads to enhanced learning capabilities because that fundamental signal of surprise and reward, the dopamine burst, it just becomes significantly stronger.
And that has, profound implications. It suggests that a really big piece of the puzzle for things like fluctuating cognitive performance or even symptoms in neuropsychiatric disorders might be tied directly to our hormonal state.
It's been under our noses the whole time.
So let's unpack the context here. We've known for decades that hormones like estrogen are incredibly powerful. They're neuromodulators.
Right. They influence everything from sleep and mood to energy levels, even certain kinds of memory.
But when you go from that sort of general influence to specific cognitive behaviors, like the rapid second-by-second updating of value that defines reinforcement learning, the exact mechanism has been, a bit of a black box.
And that's the critical knowledge gap the researchers here at Constantinople and Golden were trying to fill. They say it in their paper, that despite knowing how much these hormones affect the brain, we've known, and I'm quoting here, surprisingly little about how these hormones influence cognitive behaviors and related neurological activity.
So we knew they affected big memory centers like the hippocampus, but not how they fine-tune the actual computational algorithms of motivation.
That was the mystery. And it is not just some academic question either.
No, the clinical relevance here is huge. Why should we care about this link right now?
Well, because of what we see in human health. Look at neuropsychiatric disorders. There's so many conditions. Anxiety, major depression, even schizophrenia. They show symptoms that vary with menstrual cycles.
Right.
For some women, psychotic symptoms can get worse during specific hormonal phases, usually when estrogen is low. So if the very engine of how we learn and prioritize actions is cycling, it just makes sense that the symptoms linked to those brain circuits would cycle too.
Understanding that mechanism could open up entirely new ways to treat these conditions.
Totally. Avenues for treatment that actually account for a person's hormonal status.
Which reframes the problem completely. We'd stop looking for a single static issue and start looking for a mechanism that's, dynamically regulated.
And that brings us to the molecular star of the show, dopamine.
Let's move past the simple feel-good chemical idea. For our audience, what is its critical function in learning?
Absolutely. In the world of reinforcement learning, dopamine isn't really about pleasure. Its main job is to broadcast a teaching signal. It's called the reward prediction error, or RPD.
And that concept, the RPE, it's just so elegant. It's simply the difference between the reward you got and the reward you thought you were going to get.
Exactly. If you expected nothing and you suddenly get a huge payoff, your brain fires off a massive positive RPE, a big burst of dopamine. That signal is basically teaching your brain, hey, what you just did was way more valuable than you thought. Update your expectations.
And the opposite is true, too. If you expect a big reward and get nothing, you see a dip in dopamine, a negative RPE.
Right, which drives avoidance. And this constant signaling of RPEs is how we all, humans and animals, iteratively refine our choices to get more rewards in the future.
So the central hypothesis for this research must have been, if estrogen is influencing behavior, it has to be acting on this RPE signal.
Precisely. The question became, does estrogen act like a kind of gain control? Does it turn up the volume on that RPE teaching signal?
And if it does, how would that even show up in an animal's behavior?
To figure that out, they needed a behavioral task that was sophisticated enough to isolate that specific learning signal from just general activity or arousal.
So for their biological model, they used lab rats, and they focused on tracking the natural estrus cycle.
Which is comparable to the human reproductive cycle, but it happens much faster. This gives them these really predictable hormonal windows.
And they were looking at 2 key phases,
Yeah.
right?
Yes, 2. First, Proestrus. That's the high estrogen state. You get this sharp natural surge of 17-beta estradiol. And second, Estrus, which is the low estrogen baseline state.
And they were careful to validate this.
Oh, meticulously. They used both physical checks and blood serum measurements to confirm the hormone levels. So this reliable natural fluctuation gave them the perfect setup to test their learning hypothesis.
Okay, now let's get into the behavioral task itself. It's called the self-paced temporal wagering task. It sounds complex.
It is, but it's also brilliantly designed. The most important part is that the task is completely self-paced. The rat chooses when to start a trial.
How does it do that?
By poking its nose into a little port. And that choice, how long it waits before starting the next trial, that's the critical metric. A fast initiation time means it has high motivation, a high expected value for what's coming next.
So once the rat starts the trial, it gets an audio cue, a sound.
And that sound is the key piece of information. Its frequency tells the rat exactly how much water reward it's about to get, anywhere from 4 to 64 microliters, a guaranteed reward.
So where does the wagering part come in?
That's the clever part. The trials were organized into long blocks. And these blocks were either low reward blocks, where the average reward was pretty small, or high reward blocks, where the average reward was large.
And the rat wasn't told when the blocks changed?
Exactly. The blocks were uncued. The animal had to constantly learn and update its own internal model of the world. Am I in a good spot right now or a bad one? And it had to figure that out purely based on the pattern of recent rewards it had received.
So the goal was to see how quickly it could learn the average value of the current block it was in.
That's it. The longer the rat waited before starting the next trial, that's the trial initiation time, the lower its internal value of the current situation. If it thought the rewards were flowing, it would dive right back in, fast.
So before we even bring estrogen into it, what was the general behavior? Did it work?
It worked perfectly. Across all hormonal states, the rats behaved exactly like a reinforcement learning algorithm would predict. They started trials faster when they were anticipating bigger rewards.
So that confirms their behavior was being driven by this internal calculation of expected future reward.
Absolutely. The model held up.
Okay, now let's add the hormone. How did being in that high estrogen state, Proestrus, change this behavior compared to the low estrogen state?
The difference was,
Yeah.
it was profound and very specific. During Proestrus, when estradiol levels were high, the rats' initiation times were significantly more sensitive to those reward blocks.
Their learning rate was amplified.
It was amplified. They started trials much, much faster in the high reward blocks and much slower in the low reward blocks. They were just better at discriminating the value of their environment and responding to it.
So it wasn't just that they were more energetic or moving faster in general.
No, and that's the critical distinction.
The hormone was actually fine-tuning the calculation of value.
Exactly. If we're just generalized arousal, you'd expect them to be faster all the time across the board. But that's not what happened. The difference between their fast times and their slow times was just larger in Proestrus. They were better at tracking the shifting value.
And the computational modeling they did backed this up.
It did. The model showed that their behavior was best explained by an increase in a parameter that represents the sensitivity to the RPE signal itself.
That makes the case for enhanced learning really strong. And the data also showed a direct link between the actual amount of estrogen in their blood and this effect, right?
A direct linear correlation. The higher the measured concentration of estradiol on any given day, the more pronounced that behavioral difference was. It proved the hormone fluctuation was the driving factor behind this enhanced learning.
Okay, so we've established the what. High estrogen equals better, more sensitive reinforcement learning. Now we get to the how, the neurological how. Where is this happening in the brain?
So the researchers immediately zoomed in on the brain's central hub for RPEs and motivated behavior, the Nucleus accumbens core, or the NASC.
Right, the NACAC. It's the main target for those dopamine neurons that start in the midbrain in a place called the VTA.
It's the critical interface. It's where these RPE signals are integrated to actually influence how vigorously you behave.
In computational terms, the NALANC is kind of like the critic in the actor-critic model, right? It's the part that calculates the value of your current situation.
That's a great way to think about it. And since their behavioral measure was vigor, how fast the rat starts the next trial, and the NALANC governs vigor, it was the perfect place to look.
So to measure the dopamine signal itself, that split-second RPE burst, they needed some pretty advanced tech.
Oh yeah, cutting-edge stuff. They used an optical dopamine sensor, a tool called Grabda, and combined it with fiber photometry.
Now, why use something like that instead of, say, an older technique like microdialysis?
Well, microdialysis is great, but it only gives you a slow, averaged-out measurement of dopamine levels over minutes. It tells you about the baseline, the tonic level.
But learning happens in milliseconds.
Exactly. Learning relies on phasic dopamine, these incredibly fast bursts that happen at the precise moment of surprise. Grabda is this amazing fluorescent sensor that literally lights up when it binds to dopamine. And fiber photometry lets them thread a tiny fiber optic cable into the brain to watch that light in real time.
So they are literally watching the brain update its value map second by second.
That's what they're doing. And what they saw confirmed everything. The dopamine release in the anac core at the moment of the audio cue, it perfectly encoded that RPE signal.
A bigger than expected reward cue caused a sharp burst of dopamine.
A sharp positive burst. And a smaller than expected reward cue caused a dip in dopamine activity. It was textbook reward prediction error signaling.
Okay, so now for the multi-million-dollar neurological question. Did estrogen change this specific split-second RPE signal?
Yes, unequivocally. The phasic dopamine release in the anac core was more strongly modulated by the reward states during Proestrus, the high-estrogen phase.
So the dopamine system itself became more sensitive when estrogen was high.
It did. And here's a really crucial detail. The enhancement was specifically driven by larger, more dynamic positive RPEs.
So not all RPEs were affected equally.
No. When the rats got cues for the biggest rewards, the 32 and 64 microliter ones, the size of that dopamine burst was significantly bigger in the high estrogen state. The negative RPEs, the dips, were less affected.
That's fascinating. So high estrogen selectively cranks up the gain on the good news. It makes the big wins feel computationally even bigger.
Precisely. Which is why their computational modeling was so important. They showed that this exact behavioral pattern could be replicated mathematically by introducing a multiplicative gain factor into the learning model.
Let's talk about that term, multiplicative gain. Why is that different from just an additive gain? What's the significance?
Well, an additive gain would just shift all the signals up by the same amount.
Yeah.
A small surprise and a big surprise would both get, say, 1 extra point.
But a multiplicative gain is different.
It scales the signal based on its original size. So if the RPE is already huge, a multiplicative factor makes massive. If the RPE is small, it doesn't change it much.
Which makes perfect sense from a survival standpoint. You want your brain to learn most aggressively from the biggest, most unexpected payoffs.
And this hormonal mechanism ensures that happens. It disproportionately amplifies the very signal that tells the brain to update its value estimate upwards.
And that creates this beautiful closed loop.
It does. High estrogen increases the gain on the dopamine RPE, which leads to a bigger update of the state value, which then compels the animal to go after the next reward faster and more vigorously.
Okay, so we have the physiological proof. Estrogen boosts the size of positive RPEs, but we still need the molecular switch. How does a hormone in the blood physically change how a dopamine burst works in a synapse?
This is where they started looking at genomic changes, longer-term effects where estrogen actually changes which proteins get made. They use proteomic analysis, basically a census of all the proteins in the tissue, to compare the nanacs during high and low estrogen states.
And what did they find? What proteins were changing?
This was the breakthrough. They found that in the high estrogen states, there was a significantly reduced expression of the proteins that are responsible for clearing dopamine out of the synapse.
The cleanup crew.
The cleanup crew. Specifically, the dopamine transporter, or DEET, and also the serotonin transporter, SERT, which helps out a bit too.
Let's focus on that dopamine transporter. If the RPE is the teaching signal, the DAT is what decides how long that signal gets to stick around and teach.
It's the off switch. The DAT actively pumps dopamine back into the neuron that releases it, which terminates the signal very quickly. So if you have fewer of these transporter proteins around,
the dopamine signal is going to last longer.
It lasts longer, and that has a huge impact. When a dopamine neuron fires in a rapid burst, which is what a positive RPE is, and the DAT is slow to clean it up, those individual pulses of dopamine start to pile on top of each other.
They summate over time.
Exactly. It's called temporal summation. And it leads to a much bigger overall amplitude and a longer duration for the entire RPE signal. This one molecular mechanism reduced DAT leading to enhanced summation perfectly explains why they saw bigger, more dynamic positive RPEs during Proestrus.
So you have this clear cascade. High estrogen leads to fewer dopamine transporters, which means slower dopamine cleanup, which amplifies the RPE signal, leading to better learning and more vigor.
It's an incredibly elegant explanation for a really complex behavior. They even found another protein, SMPD3, which helps get the DD to the cell surface, was also decreased. It just adds another layer of control.
This is all very compelling evidence, but in neuroscience, you need causal proof. Did they prove that it was the inactive dopamine that caused the change in behavior?
They did. 2 really specific causal tests. First, they tackled the behavior link using optogenetics.
So they engineered the dopamine neurons so they could be controlled by light.
That's right. They could shine a precise pulse of light into the anac and artificially trigger a burst of dopamine release right when the reward cue happened.
Which is basically like faking a positive RPE. You're giving the animal a little boost of better than expected news, even if it isn't true.
Exactly. And their model predicted that if anac dopamine really drives vigor, this artificial boost should make the rats start the next trial faster.
And did it?
It did. Activating those dopamine terminals significantly decreased their trial initiation times. They got back to work more quickly. And the effect was strongest when they were in the low reward blocks, where that artificial boost made the biggest relative difference.
So that confirms it. NAVAC dopamine RPEs causally drive motivational vigor.
That settles the behavioral side. But now they had to prove that the hormone was the ultimate driver, that the estrogen receptor itself was necessary.
This was the second causal test.
The crucial second test. They targeted the estrogen receptor alpha, or ER alpha, and they knocked it down, but only in the ventral tegmental area, the VTA.
The place where the dopamine neurons are born?
Right. And this was so strategic, it proved that estrogen was acting on the dopamine source neurons themselves, telling them to change their protein expression rather than just acting locally out in the anac.
So if you get rid of the estrogen receptor in the VTA, high estrogen shouldn't be able to have its effect anymore.
Correct. And the result was a total nullification of the hormonal effect. When they knocked down ER alpha, the rat's behavioral sensitivity to the reward blocks just disappeared. Their behavior in the high estrogen phase looked exactly like their behavior in the low estrogen phase.
Wow. So that's the definitive causal chain. Estrogen receptor signaling in the midbrain is the switch that flips on enhanced reinforcement learning.
It's really a beautiful piece of science connecting all those levels of analysis.
Now, any good study has to account for other variables. The researchers did notice that when rats were in their high estrogen state, they seemed less thirsty. They did fewer trials and drank less water overall.
Right, and that's a necessary check. You have to make sure the effect you're seeing isn't just because the animals are, feeling full or conserving energy.
So how do they separate that systemic effect, the thirst, from the specific neurological effect on learning?
They use that same brilliant causal experiment. Remember, knocking down the estrogen receptor alpha in the VTA got rid of the learning enhancement. Well, crucially, that same VTA knockdown had no effect on the thirst. The rats were still less thirsty during their high estrogen phase, even though their learning was no longer enhanced.
That is a powerful dissociation. It means estrogen is doing two different things through two different pathways.
Exactly. It's influencing thirst through one mechanism, maybe in the hypothalamus, while at the same time, it's specifically targeting the VTA to fine-tune the reinforcement learning circuit. The learning boost is a dedicated neurological effect.
Okay, let's zoom out to the broader implications. This is where it gets really interesting for anyone interested in health and disease.
This mechanism has direct ties to pharmacology. The dopamine transporter and the serotonin transporter are the exact molecular targets of some of our most common psychiatric medications.
We're talking about things like DRIs, dopamine reuptake inhibitors, and SSRIs, the antidepressants.
That's right. So if estrogen is naturally turning down the number of dopamine transporters, it's essentially doing the same job as a mild dopamine reuptake inhibitor.
So what does that mean for someone who is actually taking one of those prescribed drugs?
It suggests that the baseline environment of their brain is constantly changing with their hormonal cycle. A woman taking an SSRI, for instance, might find the drug works differently or has different side effects depending on where she is in her cycle.
Because the number of available targets for that drug is fluctuating naturally.
The hormonal state is acting as this natural built-in modulator of the drug's target.
Which could explain so much of the variability we see in treatment outcomes, especially in women with depression or anxiety. When estrogen is high, it might be providing this natural cognitive buffer, this boost in motivation.
And when estrogen drops, that buffer is gone and motivation might plummet. It just demands that we move towards a more personalized approach to medicine that incorporates hormonal state.
Treating a circuit that is cycling in intensity with a static dose of medication just seems, well, suboptimal.
It really does. It encourages us to think about things like chronotherapeutics,
Mm.
maybe adjusting interventions based on documented hormonal phases.
Finally, let's just touch on the computational angle. This study really reinforces the classical role of dopamine in the anac core as this reward prediction error signal.
It does, but it adds this crucial layer of dynamic modulation. It shows that the efficacy of that teaching signal is not a fixed value.
The power of the lesson being taught, the size of that dopamine burst, is under direct biological control.
Right. And this dynamic control is something that a lot of classical static models of the brain just neglect. It's a perfect example of how hormones can directly impact the nuts and bolts of synaptic function, which in turn changes a core computational variable that we use for all learning and decision-making.
So to bring this all together, this has been a really deep dive, from behavior all the way down to regulatory proteins.
And the synthesis is pretty remarkable. Estrogen, specifically 17-beta estradiol, is a powerful and dynamic enhancer of reinforcement learning.
The mechanism is molecularly precise. High-estrogen suppresses the dopamine transporter in the nucleus accumbens,
which slows down dopamine clearance, boosts the positive reward prediction error signal,
and makes you better at learning what's valuable. And the ultimate driver is the estrogen receptor alpha, right back in the VTA where the dopamine neurons live.
So for you, the listener, I think the takeaway is to recognize the dynamic nature of your own brain. If you or someone you know experiences these cyclical changes in mood or focus or even how you respond to medication,
this research provides a potential biological reason for that variability. It's a really compelling argument for factoring hormonal state into personalized medicine.
it is.
And so we'll leave you with this to think about. If the very foundation of how we learn, how we pursue goals, how we assess risk, that core dopamine RPE signal is perpetually being tweaked by our own rhythmic hormonal cycles, how much of our motivation, our resilience, or even our vulnerability to things like addiction or mental health issues is tied to these biological realities that we've so often overlooked.
It just mandates a much more complex, much more integrated view of how the brain works. It's an incredibly exciting frontier.
It is. It's complex, it's dynamic, and that is why we do the deep dive.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.